Thanks. Yes, I'm looking forward to speaking with you all today. As you said, we're going to discuss the Ebola vaccine. And I would say that this will be an interesting story, and not so much because of when we talk about formulation development, we actually didn't get to do much formulation development of anything here. But it'll be an interesting story on the challenges that we went through. So first, we'll just go over the presentation outline, just tell you a little bit about the Ebola virus and our vaccine, and then we'll get into a little bit of the items that I think will be of interest from a drug product process development standpoint. So let's get right into it. The Ebola virus is a member of the Filoviridae family of viruses, and it was first discovered back in 1976 in the Ebola River, so that's why it's got its name, the Ebola virus. If you're not familiar with it, it causes hemorrhagic fever, and it's a deadly disease. And there's various strains, human strains. There's the Zaire, Sudan, Typhorus, and Bundibugyo. And non-human primates have the restin strain. It's an animal-borne virus, and fruit bats are considered the most likely reservoir. For this, the transmissions by direct contact, like if you have broken skin or mucous membranes with blood or bodily fluids of the infected individual or contaminated objects, as well as if you get infected fruit bats or primates. So it's a little different than with our COVID, luckily, because that the direct contact you'll see is influenced how we were able to go about tackling it in our clinical trial design. So how we got involved, like I said, 1976 was when it was really first discovered. But then there was the world's largest Ebola outbreak that was occurring like in 2014 to 2016. So the end of 2014 is when Merck became involved. We worked together with Lynne and brought the vaccine to us, and so that's when we got involved. And you can see this was the most cases that they've had. That whole epidemic was actually 28,652 cases, and there was over 11,000 deaths for that. So quite deadly. So when Merck became involved with it, it's because Lynne was smaller. We did a clinical trial evaluation, but using multiple partners along with Merck. And that's how we were able to really tackle the problem. So you can see that we were involved with the NIH, the WRAIR, we were involved with WHO. So there was multiple partnerships that made this happen and for these clinical trials. We had more than 16,000 subjects vaccinated in the phase 1, 2, and 3 trials, and that was in over 10 countries. And so this slide just emphasizes some of the numerous partnerships that we had. You see with the African governments of Canada, United States, Europe. There's universities, private sectors, and it really was everyone coming together. So remember, this was quite an effort, and I know we see things now with the COVID vaccine where we had a lot of involvement, but this to date had been the most that we had seen of multiple partners coming together. The vaccine itself is based on VSV, which is vesicular stomatitis virus. It's a recombinant virus, in which case it's replication competent, but what we have is that instead of having the outer glycoprotein, the little yellow dots around the edge is what represent the glycoproteins, it's actually switched out for the Ebola glycoprotein. And that's how we were able to generate the immune response without actually having to work with the Ebola virus itself. So the VSV glycoprotein G is replaced with the Zaire Ebola virus glycoprotein. So I think we will find interesting is that for the clinical trial, one of the main things that we had was a revaccination trial. And this again goes back to when I mentioned with the contact and the way that transmission occurs. We were able to actually focus on contacts of infected individuals and contacts of contacts. And then we evaluate these in clusters. And for the trial itself, the clusters were randomized into either immediate vaccination or delayed vaccination. At 10 days or more after randomization, We didn't have any cases of Ebola virus disease in the immediately vaccinated subjects versus 10 cases in the delayed vaccination subjects. So you see in this study, the vaccine efficacy at that point was 100%. And continued trials have been, this is how the approach goes, is always trying to get the contact and the contacts of contacts. So I think this ring vaccination approach is very interesting, has been very effective at controlling the spread of the Ebola disease. So with the outbreaks, I mentioned the one that was 2014 to 2016. There was another second largest one that occurred and that went out to 2020. And for that, we ended up with over 3,000 cases and over 2,000 deaths. But at least with this, we were able to control it much more. We vaccinated over 303,000 people. And this was supplied as part of the expanded access response in the DRC. And like I said, it was the world's second largest Ebola outbreak on record. So that was declared over on the 25th of June. Since then, there's been a couple minor outbreaks. There was one that went from May to November 2020. That was 130 people that had come down with the disease. 55 people died there. We had some in February to May of 2021. That was a small, 12 cases. And in February, June of 2021, there was 23 in Guinea. So they still do keep popping up, but luckily with the vaccine, there's a way to try and control this. So, and of course for Merck, as I think people are more familiar now after going through COVID, we get the pre-licensure support. We did stuff with emergency use, as you've heard. And then we worked licensure and also with the WHO pre-qualification, which was especially important here because of the working with a lot of the African countries where these outbreaks were occurring. And then a vaccine supply for future stockpiles. So when you get to most of the folks here are probably the drug product part. The drug product process development strategy was really trying to focus on taking what we got from Newlink and really minimize changes, move as fast as possible, and develop a commercial scale process and show comparability between clinical and commercial processes. So we didn't actually get to do the formulation development that I usually love to do due to the urgent need for the clinical trials. We just implement it right away as a frozen liquid drug product that was stored at less than or equal to minus 60 degrees Celsius. So of course this sounds easy. So what I'm going to talk about, right? It's just minus 60 liquid, away we go. But there's some unique challenges that you get for process scale up from clinical commercial when you're working with a frozen liquid vaccine, depending on the sensitivities of the vaccine. And live viruses always have some sensitivities, so. And in this case, one of the things we looked at was the impact of thaw rate on potency. And so a slow thaw rate was found to negatively impact product potency. If you look on the graph here, you're seeing relative potency. So you see fast thaw as 100% because we did normalized potency. Then relatively speaking, if it took 24-hour thaw or 48-hour thaw, you'd see around a 60% loss. And we saw those with different lots. Just to orient you, it's an activity assay. So what you're seeing, the error bars can be sort of large because that's plus or minus 2 standard error, the mean of the replicates of the potency assay. It just gives you an idea of the variability you're working with that you're trying to always struggle with infectivity assays, but to see what's truly happening there. But we did see this consistently, about a 60% loss. So what this means, and we were just looking for sensitivities. I'd like to see no change, but what it really meant then is that thawing in a 2 to 8C refrigerator is not feasible. And I think a lot of people just want to either put, whether it's in the process, they want to put their drug substance, maybe thaw on it in the refrigerator if it's frozen, or people want to just put their drug product into a refrigerator. But then that could be too slow of a thaw. So we had to rapidly thaw the drug substance for drug product manufacture to prevent this loss because it would give you a lower overall process yield. And for the drug product, we actually have you thaw at room temperature to avoid any slower thaw. Because the concern is if you're in packaging and people are throwing the whole package in, if you can extend beyond the time that we can support, because we did break this down to even some slower times. For the drug substance thaw, we rapidly thaw the commercial drug substance container and to minimize impact from the large volume, because large volumes always thaw slower, of course. So we actually implemented a water bath, and that enabled us to complete the thaw within 3 hours, and we were good to about 4 hours for what we needed to be within to not have any process loss. So I would say that GMP areas hate water baths, and you're going to have that outside of your actual, of course, your graded areas. But usually we thaw in the water bath, and you then wipe down and bring the drug substance into your processing area. Just like we looked at thaw rate, we also looked at the impact of freeze rate on potency. Luckily here, the product did not exhibit the same sensitivity to freeze rate as it did the thaw rate. If here again is relative potency, so a 15-minute freeze would be considered your fast freeze. And then we had different times, 2 hour, 8 hour, 72-hour freeze, really extending the times. And we really didn't see a sensitivity to the freeze rate. And this was very important for us freezing down the drug product, because it meant that you could just put that into the freezer and freeze it, and you would not have to worry about variability across your lot. And we actually did map the product temperature across the -70 freezer during the freezing process to confirm that all rates were within an acceptable range. For freeze-thaw stability, people often talk about freeze-thaw stability. We were actually able to go through 5 freeze-thaw cycles without really seeing any significant loss. But this was talking about working with the drug product vial. So if you take the drug product vial to thaw, you put it out on the bench top at room temperature to thaw, and then you put it into the freezer to freeze. So remember, I just said that the freeze rate didn't matter, so put it in the freezer and freeze, no issue. But the thaw rate, we pulled that drug product out. So then you can have your 5 freeze-thaw cycles. If instead people tried to do the thaw in the context if they had larger containers of vials, then it would thaw slower, and then you would have issues. So why this is important, if you think about it, when you scale it up, it's important that you couldn't do it in large quantities, because that's just going to slow down that thaw rate, and then you'd have impact and you'd have variability across your lot before you even got it out the door. So for this, I think what's interesting, we actually label before freezing for the commercial manufacturing to prevent unnecessary handling, having to worry about slow thaw times. Another thing to consider is just the warming up during handling in general. Because individual vials, and we looked at these starting out in the clinical space, because could we do anything handling individual vials? Because some places thought they could label frozen vials or handle them, but temperatures went as high as -4 degrees C within 3 minutes. So that's, although it's not completely thawed at that point, you're definitely going into the danger zone, and it wouldn't be acceptable for us. So you really need to limit the handling once frozen. So besides labeling before freezing down, we actually even packaged before freezing to prevent having to have the handling and picking up and moving vials around in any way. If you do need to do that, you have to transfer those materials on dry ice, and you also want to limit your time when you're transporting materials between freezers and locations or packing out. And this was a great struggle, I have to say, because of course everyone's like, well, we always have this amount of time. We always need this amount of time. And when we had to say that the product can't handle it, so you really need to work with the different processing areas and packaging and everything, and try and work with them to develop a system so that they can do it in the time frame that they need, but have the product temperature controlled. Another point I thought was interesting that you might find interesting here is that the stability at what is elevated temperatures for the vaccine. So remember I said it's stored at less than or equal to -60. So in this case, -20 C is elevated as well as refrigerated temperatures. So the drug product is very stable at -70 degrees C, which is its long-term storage temperature. I think we have at least 5 years of stability with the container closure integrity and everything else. But the product itself, if you put it at -20 C, you can see here, now instead of looking at potency, note that this is potency log loss on your y-axis, and then the time is across the bottom and time in months. And so basically, the higher up the value is, the worse stability is because you're losing more potency. So this is log loss versus, let's say, a -70 stored control. And you see that you really, in the first, really even 3 months at -20 C, but you lose a lot of potency. It continues to go up, but though not quite at the same rate at even out to 12 months. So it's actually less stable at -20 degrees C than at refrigerator temperatures at 2 to 8 C. So here you see at 2 to 8 C, like 12 months, on average, you're say maybe around 0.8 versus 1.6 logs. So the potency losses being greater at -20 than at 2 to 8 C, I think was very unusual for people to wrap their minds around. And so we had to keep reminding people and design our processes to make sure, like -20 C isn't where it wasn't going to be helpful to us, and actually would be better if we had to do 2 to 8. And while I'm showing you stability at different temperatures, here's another graph of stability at elevated temperature at room temperature, say at 25 C. And again, you're looking at potency log loss on your y-axis. In this case, your x-axis going across is time just in days. So remember before I was talking in months, now we're talking in days, and we're getting up to, like, in just 7 days, you're maybe like at 0.6 logs. So you see it's much more rapid loss at 25 C than at 2 to 8 C. So you weren't surprised that it's higher at 25 C, but the extent, it's much more than you would think, and you're talking just in days. So here you had to make sure that you had to control your time out of refrigeration during manufacture. And so what we did for the process there, of course, is just put everything that we possibly can at refrigerated temperatures and limit that time at room temperature. And you put those into your fill model to make sure you're building in your potencies for those to make sure your target okay. The main thing is making sure you control the process. So I think then to summarize what our DP learnings were, although the drug product was really a simple frozen liquid formulation, like I said, I didn't get to do any of my formulation development here, and that it's stored at -70, you still had to really pay careful attention to the details. It's important. I feel like we need to also remind our management and everything all the time that it's not trivial when you have this a frozen liquid. So, and then if a product exhibits stability through freeze-thaw cycling under one set of conditions, it doesn't mean it's freeze-thaw stable for all conditions. And that's very critical, like we saw. You really need to worry about the effect of freeze rate and thaw rate. These are critical. And with that, remember, volumes and positional effects are important since they impact the rates. So as you scale up and you get to those bigger quantities, depending on how they're packaged, or if you need to do a whole lot, if you wanted to try and work with a whole lot and thaw it, say the label, and then refreeze, that's where you run into trouble because of the impact they have on the rates. And of course, then a product can exhibit different sensitivity to freezing and thawing. Colder also is not always better for stability. Again, that's what we saw with the -20 C being substantially worse than the 2 to 8 C stability. And just in general, handling of a frozen liquid product after freezing requires diligent attention to transfer times and conditions. So labeling and packaging before the freeze step can help minimize this handling that's required. There's other challenges there, of course, that come into play, but I can't emphasize that enough because I think a lot of people just try and they visually look at things and aren't considering the temperature mapping and what's actually happening with the product. So that's my basically the learnings and that we had from the Ebola vaccine from the drug product perspective. I would like to mention on the current status that the product is now called Vivo. We had called it V920 as our internal project. It was actually licensed and is pre-qualified by the WHO. It had approvals in the priority African countries, which facilitated by the WHO pre-qualification. And we're looking to go into some other African countries as well. And it's also went both for the EMA and the US FDA. And finally, acknowledgements. There was a tremendous amount of people involved in all aspects of development of the Ebola vaccine, as you saw just with the partners and everything else that we had there. But I mentioned here people that were specifically involved for some of those drug product graphs and data that I showed. But there's many, many others that are contributing. So thank you.